Okay. So welcome to Toronto Data Workshop. Our guest today is Maria Kamenetsky, who's from the University of Wisconsin-Madison. Maria is going to talk about spatial clustering. She's a PhD candidate in epidemiology at Madison. And she also has a degree in statistics and also political science and economics from Madison as well. I met Maria, I guess what? Time goes on, right? It's kind of scary. It's almost 4 or 5 years ago.
Six, I think. Yeah.
Six. Oh gosh. When her and Monica were doing a data science program at Chicago, and of course they all then went back to their PhDs and took all of the data science methods to their various disciplines. In Maria's case, Epi. So very much looking forward to your presentation, Maria. Thank you very much.
Thank you for that introduction, and thank you for hosting me at the Toronto Data Workshop. Let's see, I'm going to share my slides. All right, so let's see. I think you should be able to see my slides now.
Yeah, we've got them perfect.
Okay, great. So yeah, thank you for the introduction. Today I'll be talking a little bit about spatial and spatiotemporal clustering and some methods and applications that I've been developing over the last several years. All right, so a brief overview. I'll first introduce some background and some terminology that I'll be using throughout the presentation. And then I'll introduce two methods. One is going to be a regularized approach to spatial and spatiotemporal cluster detection. And the second is going to be a stacking approach. And finally, I'll summarize with some conclusions and future work. All right, so before the pandemic, I used to have to introduce the John Snow example in more details, but now we're all experts. So I'll keep it brief. So of course, John Snow and the cholera outbreak in London in 1854, there were cholera cases, and he sort of used a map to identify where the source of the outbreak was, right? So the red points here are cases, or at least where this data set tells me the cases were, and the blue triangles are going to be water pumps. And so John Snow is a pretty famous example in spatial epidemiology. And I think that's because he really illustrates, or this example really illustrates, the power of a map, right? We don't need fancy statistical methods or advanced degrees to sort of figure out what's going on. And it really looks like most of the cases are around one single triangle, and that triangle did happen to be the Broad Street pump. And of course, once he figured that out, he triumphantly knocked down the pump and the cases went down, and we were able to sort of make a causal inference about the fact that it was that pump causing it. But yes, the point being, maps are important. And of course, now we don't have anything as dramatic as John Snow and the Broad Street pump, but we have other subtleties and other fascinating research questions that we're still trying to tackle with maps. So we're looking at how do we prevent chronic diseases, or if there are areas of elevated chronic diseases. Epidemiology, we're really interested in environmental exposures and maybe looking at cancer. Climate change and health, right? Are there pockets where air pollution or wildfires are sort of driving elevated disease risk, health disparities? Are there areas where there isn't enough access to healthcare? And of course, sort of back to the John Snow, infectious disease outbreaks and surveillance. So the map on the left-hand side, it shows age standardized county level mortality rates from breast cancer in 2014. And just by looking at this map, we can already understand a lot, again, without sort of diving into advanced statistical methods, right? There's counties that are red, so elevated risk of mortality. There are counties that are blue, right? So in Alabama, Louisiana, and then looking at sort of Arizona and New Mexico. So this is pretty easy, or not easy, but it's easy for us to sort of get patterns from this map, but that might not always be the case. All right, so before I dive any further, I want to introduce some terminology that I'm going to be using throughout the presentation. So I'm going to call cells geographic subunits that make up a study region. So we're thinking cells as like counties, states, census tracts, census blocks. I'm going to define clustering as the tendency of locations to occur more frequently in close proximity than expected. And I'm going to define a spatial cluster to be a geographic area and or time period with an elevated incidence or prevalence of disease. So for those of you who are more visual like me, I like to think in pictures. So the picture on the left is a spatial cluster. So we're really going to be talking about a lot of circles today, but we're thinking about a cluster as this area that has an elevated risk or incidence of disease. And then when we expand space to time, those circles become cylinders, and each time is going to be a slice of that cylinder. All right, so in spatial epidemiology, there are three sort of broad topic areas that people generally work in. So geographic correlation studies use geographic variation to sort of map exposures and generate hypotheses about disease etiology. Again, thinking about environmental exposures in cancer, something like that. Disease mapping summarizes spatial and spatiotemporal variation using maps of diseases. So if you're thinking about maybe some Bayesian methods, if you're familiar with INLA, a lot of that is disease mapping. A nice example is, let's say I have points and I want to map a continuous surface, a spatial surface. That'll be sort of in the disease mapping framework. And then we have our little sub area of cluster detection. So the identification of contiguous geographic areas with elevated or depressed reduced disease incidence and prevalence. So really focusing on, is this area different from the rest? All right, so back to John Snow for a little bit. So for John Snow, he had the specific cases, right? And he was able to plot them. But in modern times, we usually don't have specific geographic locations for individual people, right? Protected health information, probably, unless you're doing a case control study where you do have that, but in most health information is aggregated to some cell. So again, census tracts, census blocks, counties. So we still have those cases in sort of that transparent red points, but we're going to assume that we don't know where those cases would be. And we're going to assume that instead the information we got was aggregated to a cell. So here I've imposed a 50-meter grid, so 50-meter cells. And the information that we would get, again, maybe thinking about counties, would be, I have 10 cases in this county, or I have 10 cases in this cell. I have 4 cases in this cell. I have 1 case in this cell. And we can see that if we didn't see the points, what we would sort of find would be a little bit different from what we would be expecting, right? So the cell that has the most amount of cases is in yellow, and that's a little bit removed from where the pump was, which was a little bit more to the right. And then we have sort of our blue cells, and then we have, sort of this light green, light yellow cells as well. So this is usually the kind of data that we work with when we're thinking about health data. All right, so in many, dare I say most, public health departments, what's commonly used for cluster detection is something called the spatial scan. So we have our grid cells, and we're going to find the centroid of each grid cell, and we're going to overlay a bunch of circles. In this case, I'm just showing one circle. And what we're going to do is sort of compare what's inside that circle to what's outside that circle. And we're going to do that using a likelihood ratio test statistic. So we do this for 1 circle, 2 circles. We do this for many circles. So we have thousands upon thousands of circles. So we get a vector or a set of different likelihood ratio test statistics. And we find the one that has the maximum likelihood ratio test statistic. So we say, okay, the circle has the maximum likelihood ratio test statistic. Is it a cluster? We use a Monte Carlo based permutation test. We get a p-value. If it's less than our alpha, we say, aha, we did find our cluster. This is in fact our spatial cluster where we're expecting an elevated or reduced relative risk of disease. Okay, so this is kind of just putting a circle on the map. What does that actually mean? Well, we can sort of reinterpret that. Of all of the cells where the centroid is inside that circle, it's going to have a unique relative risk that's elevated. So here about like 1.2 or so, and everything outside of that circle is going to be 1. And so now we've identified our cluster. All right, so in the case of John Snow, in the case of looking at those age standardized mortality counts of breast cancer, visually, without doing any of this fancy spatial scan or anything, we could sort of see where there were hotspots. But that's not always going to be the case, right? So these are standardized breast cancer incidence rates for a small region in central Japan. And if we just look at a single time period, so looking at period 1, it's hard to really identify a clear pattern, right? So maybe there's something in the southern tip, maybe there's something in the top middle area. Some of these cells have an elevated relative risk. But as we then expand across time, these things change. Like, is that really a cluster? Is it not? So it's really challenging to sort of differentiate the signal from the noise in spatial cluster detection. And that really becomes the main research question of is how do we find a pattern across both space and time? All right, so a little bit of background about current methods used for identifying multiple clusters. So far I've been talking about identifying one cluster, but there might be multiple clusters in the region, right? So how do we identify many clusters? Well, the spatial scan, again, is most commonly used, and it's been extended to allow for the identification of multiple clusters. So one method is you find the cluster, you take it out, you rerun the scan. So that's sort of sequential deletion. Another approach is just formally incorporating multiple clusters in the alternative hypothesis. And then what we're going to use is another variant. There's like 100 different variants of the spatial scan if you look into it. But we're going to be looking into something we call the forward stepwise spatial scan. So it's your regular spatial scan. We're just going to update the number of expected counts when a cluster is identified. And another approach is the forward stage-wise method. So this is similar to forward stage-wise regression. So we're going to go down a gradient, and we're going to obtain a full coefficient path for each potential cluster using small incremental steps. So this is great. We are able to identify the cluster, but it's really time consuming. And it can be sensitive to the tuning of our small step size where you can get trapped in a local minima and not be able to get out of it. So these are sort of the two comparative methods that we're going to be exploring. All right, so for the two methods I'm going to be proposing, I'm going to be using the same data source. So I just want to introduce it now. So Japan is divided into 47 prefectures, and in our studies, we focus on 3 neighboring prefectures in Tochigi, Gunma, and Saitama. Across these 3 prefectures, there are 208 total cells or municipalities, and we have age standardized breast cancer incidence rates for women between, and I have to move my little thing, between 40 and 74 years old across 5 time periods from 1975 to 1994. All right, so first we create our sets of potential clusters, and we do this in the same way that we do with the spatial scan, right? And I'll go over this in a little more detail. So we have a total of capital K potential clusters. Now we have all of our municipalities or cells, right? And we find a centroid for each cell. Then centered at each centroid, we start developing circles that expand with radii from 0 up to a maximum radius that's set by the user. Now, if I have cell 1 and cell 2 and their neighbors, I'm going to say that cell 2 belongs to a potential cluster if its centroid falls within that circle. So as that circle is expanding, that potential cluster is expanding, more and more cells are members of that cluster. So for our particular study region, we set the maximum radius to 20 kilometers. And given all of this, this gives us a total of 66,870 overlapping spatiotemporal clusters, which is a lot. So next I'll just talk a little bit about the statistical model. So we assume that our observed case counts follow a Poisson distribution with rate parameter mu it as a function of the relative risk, our rho it, and EIT are the expected counts. And we have this log linear spatiotemporal cluster model. And we model the log relative risk as a function of the time varying background relative risk. So we have 5 time periods. So we're going to have 5 different background relative risks, all around 1, but with slight variability. Then we have the spatiotemporal clustering component, and I'll go through this one in detail. So theta j is going to be the log relative risk in the jth cluster. Little k is the true number of spatiotemporal clusters. And then we have this indicator function that's going to take a 1 if the distance between me, so if I'm a cell, and my center and the next center is within a certain radius. So if you think about what the indicator function is doing, it's basically creating a bunch of indicator variables or dummy variables for cluster membership. And then we have uj and lj as the upper and lower bounds of the time interval defining the temporal span. So is the cluster in time period 1, time period 1 and 2, periods 3, 4, 5. So what is the temporal span of that cluster? Okay, so we have 66,870 potential clusters. And what we do is we reframe the way we think about them, and instead we think about it in a high dimensional framework. So all of those potential clusters are binary indicator variables, and really what they are predictors. And what we want to do is perform variable selection on this. So the left-hand plot, we have the first 6 spatial clusters just across space. So as they're identified and enter the model. And then each of these has a color, which corresponds to a line on the right-hand side plot. So we can see that on the x-axis, we have our penalty term. On the y-axis, we have our coefficient estimate. So as the penalty, we start with a high penalty. So no clusters are allowed into the study region. And as the penalty gets looser and looser, more potential clusters are allowed to enter the active set, or allowed to enter the study region. So we're going to have, and we can keep doing this, again, this is just for the first several, we can keep doing this until we get 66,870 clusters in there. So just to illustrate this with a map, we'll go through the first couple. So we start again with k equals 0. The null model, no clusters are allowed into the study region. Then we have 1 cluster, 2 clusters, 3, 4, 5, 6, 7, 8, 9, 10. And I could keep going to 66,870, but I assume that you would like to eat lunch. And so I will cut it a little bit short, but we'll have, we can keep going, right? And so then the question is, okay, how many do we put into the study region, right? Like, is it 1, is it 10, is it 100? How many clusters do we want to actually put into the study region? What is being identified? And to do this, we're going to use selection criteria, and we're going to use BIC and AIC for our Poisson model. And we're going to use quasi-BIC and quasi-AIC for quasi-Poisson. So in most real life, Poisson doesn't really hold up because there are some strong assumptions, and we sort of expect there to be overdispersion in our case counts. And so we are also explore a quasi-Poisson model. And where appropriate, we're either using a log likelihood or a log quasi likelihood. Okay, so a couple of results from our simulation studies. So there's a lot, but I'll break it up piece by piece. So looking at the first 2 plots on the left where it says large population, here we're putting a cluster in a large population center. So we're thinking large observed counts, large expected counts. The 2 plots on the left, we have a detection by QBIC or BIC, and then detection by QAIC or AIC. On the x-axis of each of these, we have the relative risk inside the cluster. So we explore a relative risk of 1.1, 1.5, and 2. On the y-axis, we have percent detection. Each of the horizontal facets, we're going to have the cluster radius, so 9 kilometers, 11 kilometers, 18 kilometers. And again, looking at each of the vertical facets, we're exploring either quasi-Poisson and Poisson. Within each plot, our dashed line is going to be baseline detection. So under the null, when there are no clusters in the region, what's our detection rate, which we hope is near 0. Then power is in the light gray, and the false positive rate is in black. So again, just focusing on the 2 plots on the left for large population, we see that using QBIC, we maintain the false positive rate at or near baseline, which is what we want. We don't want to put a cluster in there if it isn't there. This is in contrast to detection by QAIC and AIC, where it really just wants to put something in there, even though the baseline detection is pretty high. So in terms of public health, even though the power is higher with QAIC, we don't want to be going to communities and telling them that we think that you might live in a cancer cluster unless we're pretty sure that there's strong enough evidence of that. So we really recommend using BIC and QBIC because it does maintain that false positive rate. Now, moving from the large population centers to the small population centers, we're now thinking about cells that have low observed counts, low expected counts. And we see that QAIC just loses its mind a little bit, like really wants to put things in there. And QBIC is still more conservative until there is enough information. So both the false positive rate and power are low until the relative risk is near 2 and the cluster has a radius of 18 kilometers. So then it will put the cluster in there. But we want this behavior. And so again, we're recommending detection by QBIC and BIC. All right, so in the beginning of the presentation, I talked a little bit about the importance of being able to detect multiple clusters. And we're going to get back to that a little bit. So if before to detect multiple clusters, we'd have to do some sort of sequential deletion, right? We find the cluster, we take it out, we rerun the algorithm. That's time consuming. So if we think about what the lasso is doing instead, we have our coefficient path, right? Those lines. And we know when each variable or each potential cluster enters the model. And so if we take a slice at any lambda value, at any penalty, we will be able to identify that in fact there are 2 clusters, 3 clusters, 5 clusters at that point. So to explore this, we develop another simulation study where we look at detection across multiple clusters. So here we put 2 clusters in a large population center with a relative risk of 2, and 1 cluster in a small population center with a relative risk of 4, all with radii of 18 kilometers, time periods 3 through 5. And we generate 100 random data sets under quasi-Poisson. Okay, so the first k equals 1, we show that this approach identifies each individual cluster, right? So there isn't bleed. You can think of it as if I identify one, then it won't identify the other. We do see a little bit of a shadowing effect in the bottom tip of the study region, and we observe this also under our null simulations. We believe this is likely just due to high observed counts. Then when it comes to k equals 2, we still successfully identify 2 clusters. Again, we still have that shadowing effect, but we do identify any combination of 2 clusters. And then finally 3, we identify all 3 clusters, and we identify it pretty well in terms of like the border between those last 2. We're not letting them bleed, which is good. Okay, and then I've talked about efficiency, so a little bit about computation time. So we compare the computation time of all 3 of our methods, all 3 of these methods under 3 different scenarios, which I call like low information, medium information, and high information. So low information corresponds to a small relative risk of 1.1, small radius of 9 kilometers. Medium information is relative risk of 1.5, 11 kilometer radius, and high information is a relative risk of 2, 18 kilometer radius. And we can see that the lasso approach is just much faster, particularly compared to forward stepwise, where the larger the more information there is, the longer it takes because it thinks that maybe there's another cluster to keep exploring. So it's a very efficient approach. All right, so finally for our real data example. So our top row are the standardized incidence rates. And then we have our results by the lasso using QBIC. So what we find is that there are 2 overlapping clusters in the southern tip of the study region. And given the overlap of the 2, this gives us 3 unique relative risks. And so we're able to sort of map a little more variability than we would otherwise. QBIC gets confused and wants to put 10 in. Using forward stagewise, it finds 5 clusters. Using AIC with forward stagewise, it finds 33, which I'm very sure is wrong. And then again, with the forward stepwise approach, it finds a single cluster in the bottom of the tip. So the good thing is all of these methods, regardless of how much it wants to put a cluster in there, it's finding the same area. So we're pretty sure there is a signal there. The question remains of, okay, what is the relative risk and how many clusters, right? Okay, so a couple of conclusions. So under both Poisson and quasi-Poisson, our lasso approach minimizes that false positive rate at baseline detection much better than any of the other approaches, and is also computationally more efficient. Once there is enough information in the cluster, it is able to also detect the cluster even in a small population center. And this is all great, but we're still limited to identifying a single correct model, right? If we're thinking about that log linear model I introduced in the beginning, we're assuming here that we have found that the small k, the true number of clusters, is 2, and this is our model. But we don't have to be limited to a single correct model. So there's been a lot of work on multi-model inference and using multiple models for that. So this is all variations of the same theme, but of course there's many different names with slight tuning. So sometimes called model mix, stacking or stacked regression, model averaging in the Bayesian framework, or superlearner. The basic idea is that you can have thousands, many models, and you're sort of going to smush the coefficient estimates using a weighted average or some sort of a weighted average. And so we believe that this approach we could also extend to spatial cluster detection, which is what we do. All right, so a little more detail about the approach. So before we had 66,870 potential clusters or covariates, right, predictors. And now we're going to reframe the way we think about it. And we're going to say that we now have 66,870 single cluster models, right? Each of those clusters is basically a single cluster model where it identifies a cluster, a potential cluster. And so what we're going to do is create a stack and sort of smush all of those cluster estimates. And I'll talk a little bit more detail about what the smushing process looks like. So for each of these single cluster models, we have a likelihood, and we're going to have a relative risk. And because we have a likelihood, we're now going to use likelihood-based weights, subject to a sum to one constraint. Okay, so visually, again, we have many thousands of circles. Each of those is now a single cluster model, and we have a relative risk, a likelihood, and a weight. So what we're going to do is we have our likelihood-based weights, and we're going to find the weight that's the highest. So we can do this either by potential cluster or by location. And we say, aha, this has the highest weight. Then we find all of the other single cluster models that overlap that location or that potential cluster. And we say, okay, this is the first ensemble of clusters that I've detected. And then we upweight everything in there to 1. And now this is the first ensemble that's been identified. Of course, this is readily extendable to identify multiple clusters as well. So we have our first ensemble detected, and we take it out by setting the likelihood everywhere there to 0. And then we reweight the remaining weights, again, subject to that sum to 1 constraint, and we repeat the process. So we're going to have ensemble 1, ensemble 2, ensemble 3, all the way until either we have no non-zero likelihoods left in our vector, if we think about it, or we set that in this study region, we're not going to have more than 15 clusters. So we'll go up to 15. So because we're going to have, again, 1 ensemble, 2 ensemble, 3 ensemble, we determine the number of ensembles, right? Or the number of clusters we want to put in our study region, again, using BIC and QBIC. Okay. So simulation studies. So one of our simulation studies, we put a spatiotemporal cluster, again, in a large population center. We give it a relative risk of 1.5, a radius of 11 kilometers, and we put it in time periods 3 through 5. And we generate, again, 100 random data sets under quasi-Poisson. So that is the top row that we see here. And we then, because we're doing model averaging, these are the maximum relative risk, average relative risk estimates inside the cluster. So all of these approaches find that cluster, which is great. But all of these approaches do not recover that relative risk. So again, the relative risk is 1.5. Stacking gets closest at 1.44. The lasso struggles a little bit, right? So if we think about what it's doing, it gets to 1.14. And then forward stage-wise and forward stepwise are sort of similar at 1.22. So the identification is similar, but now it's recovering that relative risk. All right, so again, I'll walk through these plots. So the plot on the top again is we put a cluster in a large population center, high case count, high observed counts, high expected counts. On the x-axis, we have the relative risk, where we still have the cluster relative risks of 1.1, 1.5, and 2. But in this case, we also explore a reduced cluster of 0.5 to see how well that performs. And then again, on the y-axis, we have percent detection. Each of the horizontal facets is the cluster radius, so 9 kilometers, 11 kilometers, 18 kilometers. And then each of the methods are the vertical facets. So forward stepwise really wants to put things in, has a higher false positive rate than even at baseline than we would hope for, but the power is really good. But stacking really performs most similarly to the lasso in terms of the false positive rate. It does really well, keeps the baseline detection at or near baseline. baseline, the false positive rate at or near baseline. And then power performs really well as well. And in contrast to the Lasso, it really does not struggle to get that reduced relative risk cluster as much as the Lasso does. Moving to the small population center, again, things sort of look the same. Forward stepwise really wants to find something, but even more so than with the Lasso, I think the stacking approach is able to recover a cluster in that small population center. So with an 18-kilometer radius and a relative risk of 2, we were able to recover it all of the time. In contrast to the Lasso or forward stage-wise, which had better false positive rates and better power, but not quite as good as the stacking approach. So I think there's definitely potential here, hopefully for doing some cluster detection where you don't have a lot of data or you have a lot of zeros. People in applied scientists know the plague of zeros. So yeah, I think there's definitely potential here. Okay, so our results. So there is one ensemble that we identify, and it is again in the same bottom tip of the study region. And now we're only looking at detection by QBIC. And the nice thing about stacking too is that in contrast to forward stepwise that says, okay, here's a cluster, I'm assuming the relative risk here is uniform. Stacking, due to all of the single cluster models, gives us a lot more of that spatial granularity than we would get otherwise. So these are just some summary statistics of those different relative risks. So forward stepwise says the relative risk everywhere in the cluster is 1.18. In contrast, the Lasso, right, we had 3 unique relative risks. And stacking has even more granularity. It's going to have a unique relative risk for each cell, really. Unless that happens to be the same, but more or less we can have a unique relative risk for each cell. So the maximum is the same as with the forward stepwise 1.18, but then we have a cell inside the cluster that has a relative risk of 1.01, so a border cell, right? Which we would expect. The cluster is going to have the highest relative risk, and we would expect that it goes down the further you get from the cluster center, and we hope that it's 1 outside of that cluster center. So again, we have much more of that, we capture much more of the nuance in the study region. And visually, we can see that here as well. So on the x-axis, we have the distance from the center cell in kilometers. And on the y-axis, we have the stacked relative risk estimates. And we can see that as we go from the center cell outward, the relative risk estimates quickly drop and then are set to 1, which is outside of the cluster in gray. So we have that distinct differentiation between what's inside the cluster and what's outside the cluster. And we also see that everything near the cluster core are going to be higher and similar to each other. Okay, so some conclusions. So with the stacking approach, we are no longer limited to identifying a single correct model and can use all of the information that's available to us. With the stacking relative risk estimates, we efficiently capture that fine spatial variability in the study region. And something I haven't shown you today, but we're finishing up some simulations, is because we're sort of dipping our toes into the stacking model averaging world, there are quite a few methods that allow for the calculation of confidence bounds. And so something we're finishing up working now is doing some uncertainty quantification for each of the cell-wise for each of the cell-wise stacked relative risk estimates. So we can say not only is there a cluster, but the confidence of, I have a 95% confidence interval for a center cell. So that's soon to be incorporated. All right, so some summary and future work. So again, we can successfully use the Lasso to efficiently select a parsimonious set of clusters that are different from the background relative risk in the study region. And again, stacking really helps us capture that granularity in the study region as well. All right, so for future work, there's been quite a bit of literature in the recent years on post-selection inference. So I think that some of that literature could be explored in applying, in calculating possibly confidence sets for either the cell, the specific cells, or for the cluster itself. And again, for stacking, we're finishing up incorporating some of those confidence bounds, but also the incorporation of priors on the model weights, right? So sort of going back to the more traditional model averaging Bayesian framework, we can incorporate more information into the model weights than just estimating them empirically. All right, so a quick acknowledgement to my advisors and collaborators, Ron Gagnon, Junju, and Juno Lee. And yeah, that is all I have for today. Thank you.
Thank you so much, Maria. That was just an absolute tour de force. So thank you. I really appreciate it. You answered so many questions that I had about epi that I had never been answered before. I'll just ask quickly a traditional question, and then if folks have questions, we'll stop recording and you can just raise your hand and then I'll call on you. But our first traditional question is, do you have any books that you recommend?
Yes, so for spatial epi things in particular, I like the Applied Spatial Statistics in Public Health by Waller and Gotway, I believe, and also the Statistical Methods in Spatial Epidemiology by Andrew Lawson is really good. And for just like a fun book, if you need to sort of re-believe in humanity, I just finished reading Humankind by Rutger Bregman, and that one was really good if you need to believe in the goodness of the world.
Very much. Oh, these look great. I think I like the Hopeful History one. I'll just stop recording.